This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 798, recorded on August 27th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent and everybody else. And the weather outside, it doesn't even look good. The last time we were on, it did look good, but it wasn't. This time it doesn't look good, and it isn't. It's hot and humid and sticky. But tomorrow promises to be in the 70s, and right now it's in the high 90s. So that's going to be an interesting transition to watch tonight as the thunderstorms roll through.
Dixon, it's cloudy blue skies. It's a nice day out here.
Well, go out. Have you been outside?
Yeah, I just walked back from the mouse house. It's
you
fine.
did?
It's in the 90s. It's hot, yeah. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 83 Fahrenheit, 27 Celsius. Partly cloudy, bluish sky. Just nice, warm. Fine.
Continental weather.
Also joining us from Western Massachusetts, Ellen Dove.
Good to be here. 89 Fahrenheit, 32 C, and we've got, I saw a couple of puffy cumulus clouds, and that's what turns into your isolated thunder showers. We might get a couple of those, and then supposed to be lovely tomorrow at least, 77. And
from Austin, Texas, Rich Condit.
I am not the hottest place on the podcast. We have 87 degrees, mostly sunny, but some thunderheads rolling through and making noise, but I don't think they're going to cause any damage. I have to say that once again, this has been a very mild summer in my Texas experience. We have had only 3 days over 100 degrees officially, as opposed to last year at this time around 40. Okay, so it's all good.
Yeah, but Rich, you have a hurricane heading your way, I think.
Well, it's going to miss us. It's going to make landfall like in the Mississippi River Delta. Yeah,
that's right.
So
that's right.
I think that'll mostly miss us. I wouldn't mind getting a little rain out of that, but I'm
sure
I don't feel. I'm sorry for the people who have to bear the brunt of that. It's forecast to hit as a Category 3 at this point, which is not going to be pleasant.
No.
Right.
Do we have any public service announcements? No.
Wear your mask.
Get your vaccine.
Get vaccinated. Wear your mask. Yeah. Indeed. All right, then let's jump right into it. So as many heard this week, the report on the origins of SARS-CoV-2 that Biden commissioned 3 months ago was apparently delivered to him. We don't know. We haven't seen it actually. It's supposedly being redacted.
Declassified.
They have to take the virus names out of it, I suppose.
It'll be the blank on the origins of blank.
Yeah. So anyway, we've got leaks about its. The press has speculated. I have a Times article, but there are many, many articles on this. U.S. intelligence agencies delivered a report to Biden on the virus's origins, which said we don't know where it came from. Essentially.
Yeah.
That's about the right thing to say, too.
There was a nice line in this at some point. I think it was in this article that said that some have suggested that the intelligence community is not necessarily the best crew to look for the origins of the virus. And I thought that was understated.
A very good point, yes.
Yes, that's right. That's right.
Nevertheless, the intelligence community got involved earlier on. So, okay, let's follow through and see if there's any truth to these. I can't think of a nice word. Bitlets that were dropped at the end of the last administration.
Bitlets.
Okay, and so far, nothing.
Well, and to be fair, intelligence agencies are very good at analyzing incomplete information and trying to determine what might be true and what might not be. And and they don't necessarily know the virology, but they can kind of look at it and say, well, what's this done in the past and that kind of thing. So they're not completely unqualified, but they're not experts in this.
They're also good at saying, I wonder what I would do if I were in that situation.
Yes.
They create scenarios.
Yes.
Gee, I didn't realize that your thought patterns were that evil.
Well, it's good to have people on our side who can think the evil thoughts, so we know
Exactly.
What I like is the New York Times line that is, current and former officials have repeatedly warned that finding the precise origins of the pandemic may be more of a job for scientists than spies.
Yeah.
No.
Right.
Get out
Wow,
of here.
shocking. Wow.
I can't believe that. No, I wouldn't.
Yeah.
Really. And that could take a long time,
Mm-hmm.
as you'll see.
Or never.
Took 10 years for SARS-CoV-1. So this has been in the news, and it will be again whenever this report or whatever it is. I assure you there will be no virus titers in PFU per mil in this report, which is sad because we like plaque assays. But it'll probably come out next week or in the next few weeks, and we'll have to look at it
Although,
again.
if you hold the pages at a distance, some of them may look like a plaque assay with strangely rectangular black
Yeah,
plaques.
exactly. So, but people have been asking again about this because a lot of the press coverage is framing it as they couldn't rule out a lab
Of
leak.
course, right.
Right.
Couldn't rule out.
So,
That's right.
but I thought in light of this, we would take a look at a manuscript that is in press. It's been peer reviewed and addresses much of this. It came out. Before we do that, I want to talk about a commentary that was published in Nature by members of the WHO group, the independent international members of the joint WHO China team, tasked with understanding the origin of SARS-CoV-2, 3 of whom were on TWiV, right? We had Thea Fisher-Marion Koopmans and Peter Daszak, and they're all on this opinion, as well as Vladimir Dedkov, Dominic Dwyer, Elmobasher Farag, David Heyman, Fabian Leendertz, Ken Maeda, Hung Nyun Viet, and John Watson. And they have, the title of this is Origins of SARS-CoV-2, window is closing for key scientific studies.
They don't think that the colon is the origin of SARS-CoV-2, by the way.
I put it there because you wouldn't know that there was a colon, right? By the way I read it, right? And
in virology, there's often a colon.
yes. Mm-hmm. Anyway, let's talk about this because I make some good points. In the first paragraph, they talk about this committee was tasked with understanding the origins. Report was published March, and as I have said, we had 3 members on. And, people often say the committee was not transparent. But 3 people came on and talked about pretty much all they knew. So I don't understand. Maybe that's only because 100,000 people have watched that episode or listened to it. So more of you should listen. Anyway, they write here, it was meant to be the first step in a process that has stalled. Okay.
Which they said on the show.
Yeah.
Which they said on the show.
Yes.
And the WHO has now ended this group and started a new one, which they say, oh, we don't like to say we're getting rid of this group, but they're trying to kind of reinvigorate this by treating it as a broader issue.
Well, they're making a standing committee, right?
Right. On the origins of pathogens or origins of pandemic agents or some, it's a more generic, it's not just, so it's not just SARS-CoV-2, but yeah, it's really about SARS-CoV-2.
It reminds me a lot of the revote for Brexit.
Yes.
If you don't get the right answer the first time,
right,
the election is off. We're going to try this again. Come on, everybody, wake up.
Right.
So
By the way, the episode where we had the people from this committee on was 760.
Thank you.
So they say at the end here, a WHO plans to create a committee that will oversee future origins studies. We are pleased to see this, that these outbreak investigations will be conducted routinely rather than in an ad hoc manner that could be perceived as politically motivated or with potentially punitive goals. And you can volunteer to join that committee. The deadline is September 10th, if you'd like to do that.
That's our public service announcement.
Yeah. Anyway, so they say here that the whole idea of this, so terms of reference were set up initially. It's supposed to be a collaborative study of this committee with scientists in China. It had a work plan of 8 items.
They had no input into the original terms of reference.
That's right.
Okay, so they did not set the agenda. Somebody else set the agenda for them, and they followed the agenda and added to it because the lab leak thing was not on the original agenda.
Yeah. Yeah, the possibility of a lab origin was not in the original, so it was added. And the 8 we've talked about, but they include all these retrospective studies on illnesses and serology and death certificates, mapping and tracing of the supply chains that were sold at the markets in Wuhan, testing wildlife and livestock and pets and zoo animals, and looking at the genome data and literature and so forth. And, that's gone over pretty well in that TWiV episode. And then, of course, this committee went to Wuhan in January for 28 days. And they talk a bit about what they did, right? So they say we found the laboratory origin idea they call it a hypothesis, but I don't think you should call it that. Too important to ignore. So we brought it to the discussions, and it was in the report, of course. They say we had a limited amount of time in Wuhan. I think they spent a lot of time in quarantine, right?
Yeah.
Out of those 28 days. So they said we prioritized understanding of the role of labs in the early days, biosafety procedures, potential staff illnesses, absenteeism. Spoke to leadership at the Wuhan Institute, this Chinese Center for Disease Control, and the Hubei Provincial CDC. And they say here, Chinese team was and still is reluctant to share raw data, for example, on the 174 cases identified in December 2019. And we could not get over the legal and other barriers during the time of our visit. And we decided because these cases were not likely the earliest ones, we weren't so concerned about getting them analyzed. And then it was agreed that a second phase of studies would address those cases and others.
They point out repeatedly that the goal of this study was not necessarily to solve the problem, but to set the stage. Okay, right. To define the problem and figure out best where to look. It's only phase 1.
And just to reiterate, the fact that this program of study that they're undertaking was decided before the committee was assembled. It was decided on by the WHO and the Chinese people.
Yes.
So that's where so WHO had some input into that. And also just that the Chinese reluctance to give the raw data for the 174 individuals is, they claim, because of patient confidentiality. So perhaps in the intervening time, they can figure out a way to anonymize the data and still release it and go over it.
Yeah, I think there are a couple of issues there. Patient confidentiality is certainly a totally legitimate concern. The other is the Chinese government is not super keen to relive their very first initial response in Wuhan to the virus because they've been criticized for some of the steps that they took early on, and to some extent rightly so. And I think there's a soreness there that needs to be overcome if anyone else is to see those. But as they point out in this article, those are not your index cases anyway. It didn't start then. It started earlier than that. We know that now. And if we're looking for origins, we really need to go further back than those cases anyway. So let's not get too hung up on that.
They note that it's quite clear that the virus was circulating in December 2019, but probably earlier, but they couldn't find evidence of it in their studies, and it may have been missed, although there's some genomic data that we've talked about which suggests maybe late October, early November circulation, but we don't have any clinical evidence for that yet. They also say that we didn't have any definitive proof for or against any of the 4 proposed pathways of emergence, although they did rank them, right, if you remember in the original report. And these are zoonotic infection from farm animals, zoonotic introduction through the consumption of contaminated food, or introduction through a laboratory working with animal viruses. And they say this initial report was not expected to actually answer definitively.
Right.
It was supposed to make a foundation for asking new questions for another report that they say could last months or even years. So it's still ongoing. So people say, I didn't conclude anything. Well, I would disagree with that. It wasn't
supposed to.
It wasn't supposed to, but I think it gave us a lot of information that we've covered extensively in many episodes. So please go listen to that, or else read the report. But I think if you don't do that, you can't really criticize it. They also say our critics have suggested the report dismisses the possibility of a lab leak. They put it low down on the priority, right? They said it's highly unlikely. It's more likely a zoonotic exposure to farm or other captured animals and so forth. They say we held discussions with key scientists in the relevant Wuhan institutions, a line of inquiry that exceeded our original mandate. And when we reviewed the responses to our questions, we found no evidence for leads to follow up with respect to it originating in a laboratory. The other criticism that's been leveled is that there was a focus on potential introduction of the virus through frozen food. And, this was an idea introduced from China. And they say, well, the reason we had this there, because we get in Wuhan, frozen food comes from all over the world and it's sold at the market, including frozen wild animal meat. Foodborne viral diseases, of course, happen often. And the virus can be frozen and remains infectious when thawed. So they said we had to deal with it. And I think that's completely fine. I have no issue with it.
I think that's at least as credible as the lab leak hypothesis.
Yeah, indeed.
I mean, you want to know what's possible? Well, yeah, that's totally possible.
Yeah.
I don't think it's real likely. I don't think either of those is real likely, but sure.
Yeah. And I thought this part was very good. It says, some of the public discourse around the report probably originates from miscommunication and misunderstanding about the nature of the work. And oh boy, is that true?
That's a very polite way of putting it.
Yes. It was publicly called an investigation by journalists, which isn't correct. And they say that leads to expectations that we're going to give you an answer, and it was never meant to be an investigation. So I think that communication is important. Let's see. So they mention a preprint that summarizes new data, and that's what we're going to talk about in a bit, which is now no longer a preprint. It's been peer-reviewed and it's about to be published. And it has, and we've also talked about the inventory of live mammals for sale in Wuhan markets. Remember, that was a couple of months ago, very eye-opening report, which showed that there were no bats or pangolins for sale in Wuhan at these multiple markets that they looked at.
But there was a lot of other
things. Yes. And a lot of other stuff capable of supporting coronavirus infections. And yeah.
They also talk about the deleted genome sequences, which made a big brouhaha this summer. And we had Christian Anderson and Robert Gary on to talk about that and how it didn't really mean anything. And that's what they say. The data were from people who had an onset of illness in January, so they didn't contribute anything to the origins selection, the origins question, sorry. So they say they've been meeting weekly since the publication of the joint report. And they say to keep the momentum up, they've continued to collaborate with the Chinese co-authors. And now they have put forth to WHO priorities for the phase 2 studies. And these include what they call further traceback studies, looking for early COVID-19 cases in all regions inside and outside of China that have the earliest evidence for circulation, do antibody surveys to see if infections were occurring in places that weren't reporting disease, to look at wildlife farms that supplied the markets, and to look at bats and other potential reservoirs or intermediate hosts in China and neighboring countries. And that's actually something we'll look at today.
Yes.
We have a theme in this episode, I guess.
Yeah. I've been, as I've read over the material for this, I was thinking to myself, this is going to be an episode that I will refer to many times.
Yes.
This is good.
The last section of this article is called Time's Up.
Not to be confused with the other important campaign that refers to itself with the same hashtag.
Search for the origins of SARS-CoV-2 is at a critical juncture. There's willingness to move forward from both the WHO international team and the China team. The window is closing on the biological feasibility of, looking for viruses in animals inside and outside of China.
Everybody's going to have antibodies against this soon.
And, many of the supply farms are closed. They've been culled, so it's going to be harder to see if there was a movement from a supply farm to the market. But anyway, that's basically this article. And they say the last sentence, we call on the scientific community and country leaders to join forces to expedite the phase 2 studies detailed here while there's still time. So does anyone know what is the status of these phase 2 studies? Are they going to happen?
Well, the WHO just disbanded, not supposed to use that term, but they just disbanded this group.
Really?
That did the original studies, and they're putting together this new group that has the deadline September 10th, which may, I certainly would hope, include some of the people who were on the original group. So it'll hopefully just be kind of a renaming and maybe inclusion of some more people. Mm-hmm. And a broadening of the mission. But I hope they take this phase 2 set of recommendations and agreements to heart and proceed with that. And as they point out here, this really needs to happen soon.
I do hope they include some of the same committee members, because those are the ones that are going to have established some rapport with the Chinese team already.
Yeah.
And have personal, deep insights into what's already been done.
Yeah.
I found this commentary, for anybody who wants to know what this committee was and what they did and what they found, very readable. This is really good. Mm-hmm. This is sort of the truth of what happened there.
Do we know if there's a committee in China that's doing the same thing?
Yeah, basically. I mean, there was a team in China that collaborated with these guys and that are still collaborating with these guys.
But we still get reports out of China.
Well, we're about to talk about one in this episode.
Yeah,
there you go.
We have one coming up.
Excellent.
I want to just for a moment go back to that New York Times article, because there was a sentence in this that got my back up. Okay. While many scientists were initially skeptical of the lab leak theory, at least some became more open to examining it this year. Mm-hmm. To me, the implication of that is that those who became more open to examining it were supporting the notion that it was valid.
Yep.
Okay? And that's not the case.
No.
Okay? Many of those people since have come back and said, I had no idea that I was naive in thinking that I could say, sure, we ought to look into it, and not understanding that I would be misunderstood as supporting the lab leak theory. Okay? So this sort of stuff is still going on.
Of course.
Be very careful how you read and understand, in particular, what's even in press that I ordinarily read.
No, in fact, Pamela Bjorkman, who was on the Science Letter, she wrote TWiV and she said that, what you just said. I didn't realize it was going to be used. I thought it would just prod wildlife studies. Yeah. But she said And then subsequently, other people wrote to other areas, to other places, and said the same. So
there was a classic naivete there, I think, among many of the people who commented on this.
Yeah. Anyway, so now I'd like to talk about this article, The Origins of SARS-CoV-2, a critical review, which is a pre-proof in Cell. And it was referred to in this opinion piece that we just went through as being as a preprint. Now it's been obviously reviewed and ready to be published. And from a number of people who have commented over the last year or so on Origins. The first author, there's two corresponding authors. They are bookends on the list of authors. The first is Edward Holmes, and then the last author is Andrew Rambeau, who have, genomic evolutionary biologists who have done a lot of work on this and other viruses. And then in between them, many people who have written commentaries. Susan Weiss is here, Michael Warraby, Christian Anderson, Bob Gary is on this as well, and many others from all different institutions.
And a pre-proof distinguished from a preprint. They're actually in the opposite order, you would think. So a preprint is not peer-reviewed. It's just put up. A pre-proof has been peer-reviewed and is actually closer to print than a preprint, and is now in basically the typesetting process.
All right, so the first section is called Evidence Supporting a Zoonotic Origin of SARS-CoV-2. Okay? And they go through all that, including all previous human coronavirus have zoonotic origins, as have the vast majority of human viruses. And they say the emergence of SARS-CoV-2 has signatures of these prior zoonotic events. It has similarities with SARS-CoV that went into humans in Guangdong in November 2002, and then in 2003. They were both of the emergence events that in those 2 years were associated with markets selling live animals.
Yep.
And involved species like civet cats and raccoon dogs that were sold live in Wuhan market in 2019. They're known to be susceptible to SARS-CoV-2.
This is basically a lame movie franchise. Every time around, it's the same damn thing. We've seen this before. Here it is again.
In 2003, animal traders were shown to be seropositive. They didn't have any disease. They had no diagnosis. They had high antibody levels to SARS-CoV, especially in traders specializing in civets. Then after that, in a paper that we talked about a long time ago, 3% of residents of Yunnan living close to bat caves were found to be seropositive to SARS-related coronaviruses. So basically, there's regular exposure of people in rural locations to these viruses. They say this is an important point that we'll come back to. The closest known relatives to SARS-CoV and SARS-CoV-2 are viruses from bats in Yunnan, but they say this is probably because those areas have been preferentially sampled and other areas less so. Okay, so SARS-CoV-2 is similar to the 4 endemic human coronaviruses, which we've talked about, OC43, HKU1, 229E, and NL63. And I know Kathy doesn't like that I'm reading those, but
those are names of viruses.
Those are the names of the viruses. These all have zoonotic origins. The common cold coronaviruses have zoonotic origins. We're going to talk a little bit about that in our paper, but at least 2 of them appear to involve rodents. HKU1 was first described in 2004 in Shenzhen, and it not clear where it came from, but it has a furin cleavage site in its spike
Must have been made in a
protein.
lab.
Had to be made in a lab. How can you call it a smoking gun, folks? That was originally identified in the case of human pneumonia. Okay. So I think this is important because a lot of individuals do not know this history. And when they hear it, they're like, wow, I didn't know that. And it's important, right? Because it shows the precedent for these kinds of spillovers. So as you heard from the article, the Huanan market in Wuhan was an early epicenter for infection. Some of the earliest cases there. And over 55% of cases in December 2019 had an exposure to either the Huanan or other markets in Wuhan. Most of the early cases cluster around the Huanan market, which is north of the Yangtze River. Although they say could be a matter of sampling biases. These districts were the first to have excess pneumonia deaths in January, which is how the reporting system was set up in China after SARS-1. And they say this metric is less susceptible to biases associated with case reporting. There's no epidemiological link to any other locality in Wuhan, including the Wuhan Institute of Virology, which is south of the Yangtze, and of course, the subject of speculation, as we've already told you. And
I wonder constantly whether the association with the Wuhan market really is a strong indicator that there's an intermediate animal host, or whether it just means that that's a super spreading environment because of the crowding and the people and stuff. I mean, I don't have the answer to that. This is just what I think about.
Well, there's another market too in Wuhan that had some cases. So
right.
Yeah, it could be. But I think it seems very likely that this originated through some kind of meat product, right?
Yeah.
So,
and whether there's an intermediate or not, maybe things got amplified in this market through spill back.
Yeah.
Sure. Yeah.
Sure.
In the original WHO report, they made a distinction between direct spillover and an intermediate host. And of those two possibilities, those are the two spillover possibilities. Of those two possibilities, they favored the one with the intermediate host. Unidentified.
So they say here, after the Huanan market was closed, most of the samples were destroyed, of course, as we've talked about before. But the virus, SARS-CoV-2 nucleic acid, was detected in environmental samples, like drainage areas. They did test some animal carcasses, but they're all negative, and they say these were unrepresentative of the live animal species sold, and didn't include raccoon dogs and other animals known to be susceptible to infection. So that would explain that, right?
And the context of this, I think we've touched on this in the past on TWiV, but I thought maybe we should bring it up again. And this goes all the way back to TWiV 582, which I just looked up. With January 12th, 2020, TWiV provides updates on the new coronavirus causing respiratory disease in China, current influenza season, and the epidemic of African swine fever. And this is something that developed in China from 2019 to 2020. We talked about it a couple of times on TWiV, and it had a massive impact on their food supply because they had to cull, meaning slaughter without harvesting the meat. I think it was hundreds of millions of pigs. And anybody who's ever looked at a Chinese menu knows that pork is a major protein source in the diet there. Pork prices shot up, and if you could even find it, what are you going to have for dinner? Well, let's go over to the seafood market and see what they're carrying. And so this was probably a boon to the exotic wildlife meat industry, right? You can't get pork. Maybe you can branch out into other meats. And that is all happening in the context of what we now know is this coronavirus showing up at the market. So I don't know if it was the market was more crowded or that the market had more animals in it because more people were buying them, but there was a lot of pressure on the food system at that time.
Although if you remember in that paper we did on the types of animals being sold in the Wuhan markets, they looked at the prices and they compared them to supermarket prices for chicken and pork and whatever.
Right.
And they said, this is clearly a luxury market, right? It's not where you're going to go to get your daily meal. Unless you're a billionaire, I guess.
or unless the grocery store was out of pork and you really wanted a good substitute for it.
Maybe, yeah. So who knows, right? I'm not sure we'll know that. But it's a reasonable theory, yeah. All right, so let's talk about the genomes. So viruses closely related to SARS-CoV-2 have been found in bats and in pangolins in multiple places: Southeast Asia, China, Thailand, Cambodia, Japan. We've talked about a lot of those viruses. There has been serological evidence for infection in pangolins with these viruses for over a decade. But, and this is really important, a significant evolutionary gap exists between SARS-CoV-2 and the closest known animal viruses. So the famous RATG13 is 4% genetically different from SARS-CoV-2, which is 1,150 bases to the original SARS-CoV-2 reference. That, they say, is decades of evolutionary divergence.
Let's the word they use. The word they use that works better in the public is 1,150 mutations. 1,150 changes.
Yes.
Okay, in the genome, which is a lot.
This RATG13 is less closely related to SARS-CoV-2 than we are to chimpanzees.
By a factor of 4 or something.
Yeah,
like that,
it's significant. So, it would take a long time to turn something like a chimpanzee into a human, and that's not how that works anyway. Viruses evolve a lot faster, but it's still a whole lot of evolving has to happen to get from something that looks like RATG13 to something that looks like SARS-CoV-2. They may very well have a common ancestor.
Right. Now, there are other viruses that are quite close to SARS-CoV-2. We've talked about a few of those. They have names like RMYN02, RPYN06, etc. And parts of them are closer to SARS-CoV-2 than is RATG13. And they say they probably share a more recent common ancestor with SARS-CoV-2 than RATG13. However, none of these were collected by the Wuhan Institute of Virology, and all of their genome sequences was done after the pandemic had begun. So they can't have been used to make SARS-CoV-2. None of these viruses, well, the 3 were obtained afterwards, and RATG13 is too distant. They say these data demonstrate beyond reasonable doubt that RATG13 is not the progenitor of SARS-CoV-2 with or without laboratory manipulation or experimental mutagenesis. And I can hear people saying, but, no, there are no buts.
Yeah, but they actually get to a lot of the buts in the section on the laboratory hypothesis, which I think they treat very fairly.
Yeah, we're going to go through that. Yeah, it's really worth going through because I know folks are not all going to read this.
Because they lay out all the talking points that people bring up. Oh, but this happened. Oh, but this happened. So, and they go through that, that's different from this because of this. This is different from this because of this. So
now, it is true that no bat reservoir or intermediate animal host for SARS-CoV-2 has been identified. And they say, well, probably we haven't sampled the right populations and it might be very rare and so forth. We haven't done all that much. So they say it took over a decade to discover bat viruses with over 95% similarity to SARS-CoV-2, SARS-CoV-1. And that can bind to ACE2. Over a decade.
And another point of reference here is we've talked about a few papers, and we're going to talk about another one today, where people go out and they sample a bunch of wildlife that might have coronaviruses, and they find a bunch of new coronaviruses. If we had done even a reasonable portion of the potential sampling, you wouldn't be finding new viruses every time you go out, right? There'd be a limit to this, but every one of these, it's a whole new list of viruses. Yeah, so we barely scratched the surface.
This is needle in a haystack in spades. Yes. Okay. And, as we have this discussion, I'm wondering if people, appreciate that we are swimming in viruses. Okay. There's viruses, we're surrounded by all sorts of different species. Each species has all of their own viruses. It's remarkable that the species barriers for viruses are maintained as well as they are. Okay. And there's daily, hourly opportunities for interaction between people and other species and spillover.
So 10 years for SARS-CoV-1, highly similar virus to be discovered. And they point out the animal origins of many human pathogens have not been identified. And that includes Ebola virus, hepatitis C virus, polio virus. The coronavirus is HKU1, NL63. So it's not easy. It's not easy.
So to expect an answer in 90 days.
No.
We might expect one in 90 years, but maybe not.
Or maybe 10 years if we take this seriously and really go after it.
The thing is, Kathy, most people would not get that, right? They would say, the administration is going to do an investigation. We'll find out, right? Because they don't know. They don't get it.
Because in the movie, there's always a definitive answer.
The next section is called, Could SARS-CoV-2 Have Escaped from a Laboratory? And they go through a historical recollection of laboratory incidents leading to infections, okay?
I really like that section. It's really well done.
And this is important because it's happened, although there are limitations. So SARS...
And this is a critical talking point in all of the mis and disinformation about this. It's happened so many times before. The virus has leaked out of labs. There was this incident. There was this. They list them here. We're going to get into that right up front.
So with the exception of Marburg virus, all documented laboratory escapes have been of readily identifiable viruses capable of human infection that were being worked with in high titer cell cultures in the laboratory. And Marburg virus, of course, was first picked up because a shipment of monkeys to a primate lab in Germany the monkeys happened to be harboring the virus and some of the workers got infected. Yeah, so that is the only one. Everything else. So for example, the 1977 influenza H1N1 pandemic, that was a virus that was circulating in the 1950s. We knew about it, and then that virus went away in 1968, was replaced by H3N2. Suddenly it reappeared in 1977, almost exactly the same genome as was circulating in the 50s. It probably originated from a vaccine trial, a challenge trial, they say here, although we're never going to find out where that came from. It's the only documented example of a human pandemic resulting from research activity. No epidemic has been caused by the escape of a novel virus. There are no data to suggest that the Wuhan Institute of Virology or any other laboratory was working on SARS-CoV-2 or any virus close enough to breed the progenitor prior to the pandemic. They point out
viruses. I'm sorry.
Yeah, go ahead.
I was just going to say the only viruses that escape are in novels.
Right. Yes.
Well, the ones that you know about, you're growing at high titers. That's happened before, right?
Yeah, that's true.
Or deliberately infecting people with in a challenge
trial, right? So they say here, this is important. The Wuhan Institute did viral genomic sequencing without cell culture of samples. They say this represents a negligible risk as viruses are inactivated during RNA extraction. No case of lab escape has been documented following sequencing of samples looking for viruses. All right. So what about the early days?
This is a bit that people don't quite understand. They think that, even if RATG13 is very distant from SARS-CoV-2, they still think that the Wuhan lab was actually working on the virus. No, they have sequence.
Yeah, actually,
and we went through that in a couple of different episodes.
They didn't have the virus. Zhongli, she says, we didn't have the virus. We were never able to grow it. We have the sequence. Despite extensive contact tracing of early cases during the COVID-19 pandemic, no reported cases related to any laboratory staff at Wuhan Institute of Virology, any staff in Dr. Shi Zhongli's lab. They're all seronegative when tested in March 2020, and they followed the appropriate biosafety protocols for coronavirus research. These are all things that people are saying, it was an outbreak in the lab. They were violating biosafety. No, they did all the right thing. Let's see. Oh, this is a very good point here. Modeling suggests that the number of hypothetical cases needed to result in multiple hospitalized COVID-19 patients prior to December 2019 is incompatible with observed clinical genomic and epidemiological data. So people say, there were sick people and we didn't detect them, or they were. But no, if there were, there would be many, many more people infected, and there's no evidence for that, right?
Yeah.
So that's important epidemiological modeling data that goes to support that.
Now, what about the Wuhan Institute of Virology? What did they have? This is another issue that's discussed. And it's discussed when people don't know anything about what they had. So here now we're going to find out. They had a lot of samples from bats, samples derived from bats, which they have published. They have isolated in cell culture 3 SARS-related coronaviruses from bats. They're called WIV1, WIV16, and RS4874. All 3 of these are more closely related to SARS-CoV than SARS-CoV-2. The RATG13 from the Wuhan Institute was never isolated nor cultured and exists only as a nucleotide sequence assembled from short sequencing reads. Okay? They don't have the virus RATG13, which...
They don't even have the long, full-length genome that you might imagine could get transfected or somehow taken into a cell. It's short pieces of sequence.
They've got the plans for it that have been put through a shredder. Basically,
right.
They've got fragmented RNA that they were able to sequence and reassemble and say, this is the sequence this would have come from, put the sequence in the database, but you don't get virus out of that.
And it's still 1,000 changes short of
that still would only get you to RATG13.
So these 3 viruses that they have isolated from bats, WIV1, et cetera, they were all isolated by passage of material through viro cells, vervet green monkey kidney cells, which they say a process that consistently results in the loss of the furin cleavage site. So if you said, well, they passed the ancestor of SARS-CoV-2 in the lab and it became SARS-CoV-2, not likely, because SARS-CoV-2 has a furin cleavage site. We're going to talk more about that in a bit. But the way it would have been done based on the history of the lab wouldn't have a furin cleavage site. They have done some work on recombinant coronaviruses, which we've talked about. They've used as the genetic backbone, WIV1. And these studies, also done in Ralph Barrick's lab, they took spike mRNA sequences from various bat-related coronas, and they said, can these bind ACE2? So they put them in WIV genome and then infect cells and see if they can get in via ACE2. And some people have said, well, that led to SARS-CoV-2, but no, WIV1 is not related to SARS-CoV-2 and can't be. And they also say SARS-CoV-2 has no evidence of genetic markers one might expect from laboratory experiments.
And I think this next sentence is really critical. There's no rational experimental reason why a new genetic system would be developed using an unknown and unpublished virus with no evidence nor mention of SARS-CoV-2-like virus in any prior publication or study from the WIV. You wouldn't take something you'd never worked on before and say, hey, let's do a gain-of-function study. What's even your hypothesis there? You don't even know what this thing is. There are steps to go through, and you would publish those steps, and none of that happened.
There's also no evidence that the Institute sequenced a virus that's closer to SARS-CoV-2, right? She's been asked. She said, no, we didn't have anything closer. No reason to hide research on a SARS-CoV-2-like virus prior to the pandemic. Under any laboratory escape scenario, SARS-CoV-2 would have to have been present in a lab prior to the pandemic, yet no evidence exists to support such a notion. I'm glad they call it a notion. And no sequence has been identified that could have served as a precursor. So someone sent me an email this week. There are a few scientists, otherwise reputable, who are very outspoken against gain of function research. Right? I don't want to mention their names because I don't want to be nasty to anyone. But one of them said somewhere they used a new ligation method that erases all evidence of manipulation to make this virus. One of these supposedly reputable scientists said that. Do you know of any ligation method that erases all evidence of manipulation?
Or why you would want such a method that would mess up all your subsequent experiments? No.
It's crazy. It's just the stuff people think up. And so someone said to me, is that possible? I said, they're just throwing words at you to make you think they know what they're talking about. And you say, it must be true. It sounds pretty good, right?
Well, they've got the little microchip in the vaccine, too. Don't forget that. So those are the same people. No, maybe not this one in particular.
Okay, so another idea has been, all right, so they're working, even though there's no evidence they had any virus close to SARS-CoV-2. Okay, let's humor you. And a lab escape scenario involves accidental infection in the course of serial passage of some related coronavirus in lab animals. Say mice, right? Most likely you'd be using mice.
Because they're cheap.
However, the early SARS-CoV-2 isolates didn't infect wild-type mice. There had to be changes. And in fact, when they finally are able to infect them, they get mild disease. And they say the fact that the early virus could not infect mice is inconsistent with a virus selected for pathogenicity and transmissibility through serial passage through rodents. Okay, so let's listen to that again. The original SARS-CoV-2 isolates up to a certain point could not infect mice. So you're saying it was created by passage through mice. Maybe you need to step back and think about your logic.
Why is it so bad at passage through mice if it was created by passage?
Yes.
And then you'd have to invoke, well, maybe they use some other animal, but that doesn't, now you're getting way out there. Yeah, nobody does passaging experiments in monkeys.
Like snow leopards.
Or ferrets.
Or ferrets, too. They're too expensive, yeah. And now we know subsequently that several changes in spike that have arisen naturally in humans and other animals, like N501Y, they can allow infection of mice. But we don't see any of those markers in the early isolates. They say low pathogenicity, absence of genomic markers associated with rodent adaptation indicate that SARS-CoV-2 is highly unlikely to have been acquired by laboratory workers in the course of experiments.
I want to point out as we go through this, I've kind of already said this, but these are not just straw men that the authors are bringing up and then knocking down to make it look like they're creating an argument. These are arguments that other people have levied to support the lab origin notion. Yes. Okay.
They're being presented in their most credible form.
Right.
Like, here's all the evidence supporting this. And then here's why that doesn't work.
All right. Then the next section is called Evidence from Genomic Structure and Ongoing Evolution of SARS-CoV-2. And they say here that, all right, so as we have said, there's claims that the virus was genetically engineered to promote human transmission. However, since we found SARS-CoV-2, it's experienced repeated sweeps of mutations that have increased viral fitness, right? It's getting fitter. Now, wait, you told me you made it really fit before you released it into the world or accidentally released it, but it's still getting better. And so they go through some of these changes, in the receptor binder domain that have been selected for apparently as it's passed through the human population. They say these refute claims that the spike was optimized for binding because it's getting better and better as these changes emerge. They also say that some pangolin-derived coronas have binding domains that bind even better to ACE2 than SARS-CoV-2.
And I really appreciate this argument that the promiscuous host range is a real argument against this, because if you had done any effort to adapt this to human hosts, you would not probably get a virus that is so easily transmitted to mink and cats and everything people look at. It seems it's all over the place.
Yeah, they call it a host generalist.
Host
virus.
generalist.
God,
Yes.
I don't remember that term being used. We used to call it broad tropism, right? Or something.
Or promiscuous.
Yeah. Species specific. Anyway, they say these findings show no specific human in quotes, adaptation was required for the early spread, and the claim that the virus was already highly adapted or somehow optimized for binding to ACE2 is without validity. Now, how about the furin cleavage site? They note this has been subject to recurrent speculation. That's just being very calm, isn't it?
Very nicely put. Yes.
All right. So they know this furin cleavage site is not present in the closest known relatives, but it's perfectly reasonable because we've barely sampled the lineages. We have less than 10 isolates that are really close to it. But the furin cleavage sites are commonplace in other coronavirus spike proteins, feline alpha coronaviruses, MERS coronaviruses, some strains of mouse hepatitis virus, endemic human beta coronaviruses like OC43 and HKU1. In fact, there is a near identical nucleotide sequence in the spike gene of the bat coronavirus HKU9-1. And if you look at the sequences, there are sequences near this furin cleavage site that are indicative that recombination has taken place to perhaps introduce the furin cleavage site. So they say simple evolutionary mechanisms can readily explain the evolution of an out-of-frame insertion of a furin cleavage site. What? Out of frame? What does that mean? Actually, Bob Gary talked a lot about that as well. So the SARS-CoV-2 furin cleavage site, RRAR, arginine, arginine, alanine, arginine, right? Doesn't match the canonical sequence, which would be RX, any amino acid, either R or K, lysine, and then R. And it's suboptimal compared to the furin cleavage sites of HKU1 and OC43. And it was caused by an out-of-frame insertion. Okay? This is the good sentence. There is no logical reason why an engineered virus would utilize such a suboptimal furin cleavage site, which would entail such an unusual and needlessly complex feat of genetic engineering. You would put it in, not just suboptimal, but you'd put it in out of frame. Your advisor would say, go back and redo it. Why did you do this? I'm sorry, I wasn't thinking clearly. And they say nobody at the Institute of Virology in Wuhan was ever doing this kind of stuff, putting furin cleavage sites into coronaviruses. There's no evidence for anybody doing that. A couple more things about the furin cleavage site. There are some substitutions upstream of the furin cleavage site that are seen in circulating viruses that improve cleavage of the spike, and they say is another signature of ongoing human adaptation. And also the SARS-CoV-2 furin site is lost if you grow the virus in viro cells in culture. So they probably would have used those cells because that's what they use for all their other viruses.
That was their standard procedure.
Yeah. Now, it has been claimed, again by one of these otherwise reputable scientists, that the presence of two adjacent CGG codons for arginine is clearly a site of human engineering. But who would put two CGG codons in a row? But they say that's not true. These codons are rare, but it's found throughout the genome of not just SARS-CoV-2, but SARS-CoV and other human coronaviruses at similar frequencies. And they say,
who put them in all those viruses?
Yes. And they say if you had put a low fitness codon into the virus, it would have been quickly selected against during the passage of the virus in the human population. Yet these CGG codons are over 99.8% conserved among the over 2.3 million near complete SARS-CoV-2 genome sequences that we have. Wow. 2.3 million nearly complete.
That's a lot.
That is a lot. Indicative of strong functional constraints. So the CGG is maintained, and there's a reason for it. We don't know what it is. We'd like to know.
Yeah.
But with the knowledge that it's rare, you probably wouldn't have used that codon if you were putting arginines in. That's the point also. All right, so that's it. The conclusions. The most parsimonious explanation is a zoonotic event. What does parsimonious mean? Should we explain that?
Simplest.
Thank you, Kathy. Good.
Most parsimonious. Simplest is a zoonotic event. And they summarize basically what we have just told you, and why it's not the history of lab spillovers and so forth. There is no evidence that SARS-CoV-2 has a lab origin. But there are clear epidemiological links to animal markets. So why? And people always say, well, why did it arise in a city where there's an institute studying coronaviruses? Well, it's coincidence. It's a big city, and probably a big city with a lot of people and a lot of travel. It's a really well-connected city. Maybe that's important for it to spill over, right?
Yeah,
And
this final sentence, keep this in mind as we go through the paper for today. But the link to Wuhan therefore more likely reflects the fact that pathogens often require heavily populated areas to become established. And it's something that, Vincent, I know you and I teach in our classes about measles and smallpox needing larger centers of population in order to survive and thrive and be carried forward as pathogens.
And you can also reverse the question, why is the Wuhan Institute of Virology in Wuhan? Because they wanted, they were, looking at places to put it, and they waited for a big city that, has a little history here.
Yeah, it's highly internationally connected. And for science, you want to be connected, right?
Yeah.
So I can hear the skeptics in my mind saying, well, you can't trust the scientists, okay? They're just trying to cover their butts, okay, and protect the other scientists who leaked this. So we have 21 authors here that are some of the most reputable scientists with the most relevant experience in the world, representing 7 different countries: Australia, US, Canada, UK, New Zealand, China, and Austria. So if the scientists are collaborating to cover their butts, this is an amazing conspiracy, and it's global. All right. I should note also that there were 11 different authors and members of the original team that the WHO put together. Similarly, a global representation and experts in the field. So I would levy the same argument. Okay, dude, pay attention.
I had a postdoc join my lab in, I think it was around 96, and he was from China. And I remember Lois Miller, a virologist in my department, commenting on the fact that, oh, you got your training in Wuhan. What a really great city for training in virology. So it really predates any of this SARS coronavirus stuff.
That's the only reason this theory has any kind of legs at all, because the Wuhan Institute of Virology is there. Because anywhere else, nobody would be talking about this.
If this had shown up in Guangzhou, we would not be having this conversation over and over again.
Which is where SARS-1 showed up, and nobody thought about it, right?
Right.
So I think it's just, I personally, as a virologist, I'm just stunned that anyone is thinking about it. And in fact, they say here, and I think this sentence, I would not have put this in. While the possibility of a lab accident cannot be entirely dismissed. I think in this case, it can be. We just heard maybe others can't be dismissed. But in this case, I think you can dismiss it. They just say it's highly unlikely relative to what we know. Why can't you dismiss it?
You can't disprove it. It's not a viable hypothesis, as you correctly pointed out, because you can't disprove it.
You can't prove a negative.
No,
that's the problem.
I could dismiss it. Sure.
Yes. You can dismiss considering it because there is no reasonable evidence to support it.
All right. So they should have used disprove then instead of dismiss, right? Okay. That's fine. Failure to comprehensively investigate the zoonotic origin through collaborative and carefully coordinated studies would leave the world vulnerable to future pandemics arising from the same human activities that have repeatedly put us on a collision course with novel viruses.
That's the biggest problem. This is a huge distraction.
Yes.
Okay.
Well, I think it's pissing China off, right? And
Of
they're
course
saying,
it is.
Yeah.
screw you.
Yeah.
You can't have any of our records, right?
Absolutely.
And in
How
a
do
way,
you think the CDC would respond if there'd been, some pandemic that originated in Atlanta and China came over and was browbeating us to turn over all of our data?
anyway,
I think we've got a phrase for that.
Dixon, are you convinced?
You are muted.
Just trying to wake Dixon up here.
Of course I was always convinced.
Okay.
You follow the science. That's what you do. You follow the science.
You're just such a scientist, Dixon.
At any rate, another
Retired scientist.
another very readable paper.
Yes.
Okay, all these links for this episode, I think anybody could pick up and make sense of.
They're all open access and they're all highly readable.
And it's really nice because it's comprehensive.
Yeah.
Both the WHO commentary and this are really comprehensive, and they're from the people who know what's going on.
They have a couple of figures too.
Yep, great stuff.
Okay. Now, related. Oh, before that, yes. Before that. So as you know, the Pfizer vaccine, mRNA vaccine for COVID-19 was given licensure this week by the FDA, right? And so with that has a name. They gave it a name. It's no longer BNT, whatever. It's Comirnaty. Just rolls off the tongue.
Comirnaty or
Comirnaty.
Comirnaty.
Oh, you know
Comirnaty.
how to pronounce it because it's a real word, right?
Well, they
gave the pronunciation.
How
How the hell would they know? It's a fake word. You can pronounce
They
it.
gave a rationale and a pronunciation.
I know what the rationale is, Dr. Spindler, but you could pronounce it any bloody way you want because it's a fake word. I'm sorry. And if somebody looks at it and says Comirnaty or whatever it is, it doesn't matter in my book. But
it's a horrible name, but in their defense, I really kind of feel for the folks at pharma companies who have to come up with these things because they're running out. You've got to have a name that's relatively short. It's got to be memorable. It ideally would make some kind of sense to doctors. Since we have this this pathology in the United States called direct-to-consumer prescription drug advertising, you also want it to be comprehensible or at least pronounceable to the lay public. So you've got all these constraints on it, and then so many names are already taken. I mean, between prescription drugs and Pokemon, we're running out of stuff that would fit this, and so
I found
ask
a nice
your doctor about Aerodactyl.
I found a nice article that goes through what Alan has just said, and they did have a pronunciation guide in the release. Yeah. Comirnaty. And of course, it's supposed to mean what? COVID-19 immunity and mRNA.
That's right.
Right.
That's right.
Yeah.
That's right. That's
And
right.
they
Plus, it conjures up the community.
Community,
idea.
yes.
Yeah.
Does it conjure community to you guys?
Sure. If you say, if you pronounce it right.
If you
Vincent.
pronounce it right, it does.
Comirnaty. So the mir part, microRNA, right? That I always liked the mir 120, the microRNA numbering.
But they've added in the I
Yeah,
just to make it
pronounceable.
It's
Yeah.
mRNA. And so I think it's really Comirnaty. Which sounds a
Comirnaty.
lot more like, don't say it mir,
Comirnaty.
Comirnaty.
So the pronunciation, Kathy, did you see this article that I see that the KOE-MIR with the apostrophe?
Right. The apostrophe tells you the accent.
Yeah, the emphasis.
Syllable to put the accent on.
But it doesn't tell you to put the mir as opposed to mir, though, right? That's
Because they didn't use IPA. If they used IPA, it would be unquestionable.
But it would also be unreadable by most people.
Exactly.
all right. So let's practice, folks. Comirnaty.
Comirnaty.
You have to
I got it.
Comirnaty. Okay. Now there's another vaccine.
Before transferring to a 4-year institution, many people go to a community college.
This is all true.
So there are some other names. So they registered this name in June 2020. Pfizer did. Wow. So they're thinking about it.
Because they didn't want anybody to beat them to.
They also BioNTech trademarked a few other names. Covuity.
Probably covuity, but yeah.
Covuity, and then Covimyrna. And Covimyrna.
The K on the beginning of that makes it look Russian.
Or RNX Covey. How would you say RNX? Would you say RNX or RNX Covey? What do you think?
I wouldn't say it.
And then some other
Covey. Too many syllables.
By the way, did you know that Moderna has applied the trademark Spikevax? SPYKE.
That's a good one.
I like that.
That's a good one.
Yeah.
But why the Y though? Why not? You think someone else had the I?
Yes.
Could be.
Maybe.
And they wouldn't sell
So,
it.
Alan, if they came up with a Pokemon name, are those all out of consideration?
I think they would be for drug names because, first of all, there would be the intellectual property issues that Niantic might want to have a word with you, and then more importantly, I think there'd be the issue of confusion and ridicule, of course.
Yeah.
Because
So
I searched
TV and my health has evolved. Yeah.
And if you look at car names, it's the same deal, right?
Sure.
Oh yeah.
Any brand name.
Yeah, that's another. It's not just Pokemon; it's cars that take a lot of
It's
it.
cars.
Yeah, and you gotta make sure that it's not an insult or a dirty word in some other language.
That too.
This is all
right.
true. This is all
Matt,
true.
Kathy, what were you going to say?
Well, I was just going to say I searched the Pokemon names and none of them have mRNA in them. So
Right.
That's too bad.
It could be possible to have come up with a new Pokemon.
It could be.
So speaking of cars,
They're all DNA characters.
speaking of cars, someone sent me an email and said, "Did you know that now there's a virus car, the LaCrosse? What is it? Buick LaCrosse?
Yeah, that's been
always
around for a while.
They've had one. Yes,
Isn't that funny?
that's been around
So
a
then
while.
there's this AstraZeneca name, Vaxzevria. Wow. I don't know. I like Covishield. I think that's great.
Covishield is very good.
Covishield girl.
Yeah, but words like that would be perilously close to things like Kovacs and
Yes.
other terms that are already used. So they talk
Yeah.
about the fact that you have to avoid
Yep.
putting in the name the key ingredients in a way that makes people think that's the only active ingredient. And
Right.
there are a lot of rules,
Yeah.
that they
Yeah.
have to.
And you have to make sure it's not too similar to another drug or vaccine name, because then you get screw ups in prescriptions and communication in clinical settings.
Right.
So the FDA has a 42-page document on how to name drugs and vaccines. Should we discuss that on a future episode?
No.
No.
Well, but you see patterns. Like anything that's a monoclonal antibody is going to have that MAB in the
AB.
name.
Yeah, that's MAB.
Yeah.
That's
But there's no pattern for vaccines, right?
not yet.
I don't think so.
Maybe there should be.
Not yet.
What's the
There are
name
only
of the...
so many vax words you can get.
So
though.
is OPV the official name of oral polio vaccine, Alan? Do you know?
I don't remember. It's not. I think it's not that simple. I don't know.
All I can think of is Flu Mist.
Flumist.
Right.
What I think is a trade name.
That's a
but-
great name. Flumist is wonderful, right?
But all the flu vaccines have trade names.
I like Shingrix is good, right?
Shingrix is excellent.
Because it's got the disease in there. But then you have something like acellular pertussis, right? Which is really a
They
science.
probably all have names. We just don't know what they are.
Yeah, I don't. That's what I'm trying to see here. What's the name of the polio vaccine? What would you call it? The trade name?
Yes.
Yeah, probably.
Trade name of polio. I mean, you could look. Oh, look at this. IPOL. It's called IPOL, polio vaccine inactivate. IPOL is the name.
That's the inactivated one.
It's the one made by Sanofi, the inactivated one. Yeah, interesting. Wow.
In contrast to Interpol.
So the CDC has a whole list of vaccine names. What would you like to know?
Opviro, I think, is oral polio vaccine.
Look at this. Look at this
Measles.
list. IPOL is the IPV. We don't have that.
What's measles?
Measles? Measles,
That's
mumps,
part of-
rubella is called
Yeah.
MMR.
Just MMR.
MMR.
You can put the link in the show notes for
This
everybody.
is
who's
very
curious.
nice. There's some here. I'm going to tell you some names, and you guess what virus they are.
Or car,
There.
or Pokemon.
Okay, you
In fact,
ready?
I think I saw an online quiz
Folks,
that was
we
like,
might
We're all
get
gonna
to
fail this
today's
exam, you
paper.
know.
we might get to today's paper eventually.
Yeah, we will. How about Imovax? Do you know what that is?
Oh, I've heard of that. I call it Imavax.
Imavax. I pronounce everything wrong.
What the heck is that? What the heck is? I don't know.
It's a rabies vaccine.
Oh.
How about Rotarix?
Oh, that's rotavirus.
Yeah, I gave you an easy one.
No brainer.
How about ACAM 2000?
Oh, pox, dude.
How about
Pox?
YF Vax?
That's yellow fever.
Yellow fever.
Oh, you guys are brilliant. How about Ixiaro? All right, let me pronounce it a better way. Ixiaro. Do you hear my Japanese accent?
Was that a Mayan
Japanese
god of?
Don't tell me Japanese encephalitis. No.
You got it.
Really?
Yeah.
Ixiaro.
Ixiaro. And the flu ones all have flu in the name. Here's an adenovirus vaccine, Kathy. This is hard. Are you ready? Adenovirus type 4 and type 7. Okay. Wow.
Vincent, could you round those numbers off for us, please?
That's very nice. I love this discovery. Let me put that in here. I didn't know
What
this.
is in a name? Let me tell you.
All right, now we have a paper. It is a JV Journal of Virology accepted manuscript. It is a novel, potentially recombinant rodent coronavirus with a polybasic cleavage site in the spike protein. And this comes from a variety of scientists over in China. And the first author is
4 of them.
Xin Li, Liang Wang, Pei Pei Liu, and Hongying Li. The corresponding author is George Gao.
And William Liu.
And he's also a corresponding. And Peter Daszak is on here. And Kathy, do you want to give us the summary up front?
I can give you a summary. It may not be
May
that
not
short,
be summary.
But anyway, what these authors did was collect 300 or so rodents from urban and rural sites in southern China. And they found that the overall prevalence was about 12%, but it was very biased toward Guangzhou, where there were 23% of the positive cases. And only
Prevalence of coronavirus.
0.7% in Guilin. And then in another area where they sampled rodents, they got none. A couple of interesting facts were that in the rodents from urban areas, there were much more higher levels of coronavirus prevalence than in the rural areas. And the urban sites with the highest proportions of rodents were passenger stations and hotels in Guangzhou. So stay away from the public transportation and hotels.
I've been in Guangzhou in hotels and passengers. Nothing bad happened to me, but
Did you see any rats?
I think so. Yeah, we saw some.
So the bottom line is that while we may think a lot about bats, we also should be thinking about effective surveillance and of other mammals such as rodents. And they kind of come down on the side of what we call One Health, which is thinking about animal health in order to think about human health.
We've already discussed the fact that this SARS-CoV-2 is a generalist virus. Okay, it seems to get around, can spill back, spill over again, and there are endemic human coronaviruses that supposedly came from rats. So, all the focus on bats is probably too narrow. Let's have a look around.
And this species promiscuity is not unique to SARS-CoV-2, even just among coronaviruses. This is a group of viruses that can often get between species maybe a little more easily than some other groups of viruses.
And then among the 30-some positive animals that they found out of about 300, they focus in on one that seems to be a completely new coronavirus based on it has enough sequence divergence to be classified as something new. And they even postulate that it could have arisen through some kind of recombination, and they show that those results.
So bats are 20% of mammals, but did you know rodents are 40% of mammals?
Yes.
Dixon, you knew that?
Yes, I did actually.
Because in the past, Dixon, you have always, crowed that bats are 20%, but you never mentioned the rodents.
Crows are not nearly as diverse.
No, they're not. That's true. No, I just found that I thought bats were rather exotic until I heard that figure. That's all. I knew that rodents were not exotic.
I know some rodents that would dispute that
think?
bats are exotic. Yes, nevertheless, yeah. So this is about giving rodents their
They're
little
due.
spotlight,
That's
right?
That's right.
And by the
And
way,
they tend to be in at least a few species of them are often in very close contact with humans.
that's right.
As this discusses.
Very domestic.
Yes.
And circling back to what Rich said earlier about looking for a needle in a haystack, if you're only looking in the bat haystack, you might not find the origins of SARS-CoV-2 even.
Looking beyond the bat haystack.
That's right.
And can anyone tell me what human viral pathogens originated in rodents?
OC43?
Yeah, OC43 and HKU1. But how about outside the coronaviruses?
Hantavirus.
Hantaviruses. And you could tell another virology teacher.
That's right, Hanta. Exactly.
Well, yeah.
Hunt. Any others though? That's the thing. Yes, there are certainly. There are hemorrhagic fevers all over the place that originally. I'm thinking of Argentine Junin hemorrhagic fever. That's a rodent in the Argentine pampa, and there are others. There are many others as well. Rodent-associated viruses, but rodents are important.
Yeah. And rodent-associated bacterial infections too, infamously.
I spent the afternoon or the morning with rodents here. Yes. Some of my finer friends. And they do live close to us. They do live close to us. But the wild ones are so fast compared to lab mice. It's just amazing. All right. So you've heard this paper. Basically, they conducted sampling in southern China. And they did both urban and rural areas, right? So you've heard the passenger stations, the hotels. Can you imagine going into a hotel? We'd like to catch some of your rats.
Hey, help yourself.
Well, there's at least more than, I think, one hotel where there were no rats collected. So,
and then they have wildlife farms where they raise animals for food. Free-ranging.
They don't name the hotels, though.
No, they don't.
So
I don't think the hotels don't want to be named.
They would not appreciate being named. It's why the CDC, and anytime you read something in MMWR that happened at some restaurant, so we visited restaurant A.
They captured free-ranging rodents in shrews using metal traps. And then they did rectal and oral swabs from all captured individuals. And they said sometimes they died, so we got their intestinal specimens as well. Accidental death. And then they extract the RNA and they do RT-PCR using degenerate primers that would pick up any coronavirus, essentially. So they're focused on the coronaviruses.
I just had one question for everybody. I had the impression that this was catch and release because they only did the oral and anal swabs. And then from a small sample, they were able to get intestinal samples, but they specifically say either they were dead or they died during handling.
Accidental death, yeah.
So, but they never say, and we released all these other ones, but I assumed that was the case.
Maybe they ate them.
Well, or just euthanized them. It seemed interesting to me, actually, that, if you're catching, especially if you're in a hotel and you're catching their rats and you're going to release the rats.
Back in the hotel?
Do you release them back into the hotel?
No, probably not. I think you release them.
I mean, at least confiscate their keys.
You release them across the street and then they come back to the hotel later.
Then they come back to the hotel that they were in, of course, yeah.
There are so many rats
through the subway
in some places that they're eaten seasonally as a delicacy. And if you go to India, which I've had the privilege of going to several times now, the rat is a revered animal because it holds up Ganesh, which is the elephant god. Yeah. And the platform that Ganesh sits on is being levitated by
rodents, rats.
A horde of rats. So there's a conflict.
Yeah.
between public health and religion in this sense, because they do have rat collectors
sure.
that go along and they try to get rid of as many as they can. But it's like trying to get rid of all the monkeys in the temples of India. You can't. It's just so many that you'll never do it. It's just never going to happen.
Dixon, the rats in this study were Rattus norvegicus, the brown.
That's the Norwegian rat.
Is that the one that bit you when you were young?
Probably. Yeah, the wharf rat. It's known as the wharf rat also.
It has a bunch of names.
Yeah.
It's the ubiquitous rat. It's when people talk about rat infestations, this is the rat.
So they captured nearly 300 rodents, Kathy.
Gee.
Nearly 300.
Yeah, that's the rest
Nice
of it
round
much
number.
too fast.
Right.
35 of those that's about 10%, right? Roughly.
Yep. Roughly 10%.
Were coronavirus positive. Not actually coronavirus positive, but coronavirus RNA positive.
No, that's right.
Yes.
That's right.
And
They did try to culture virus.
it failed, right?
Yeah. But so they must listen to TWiV. They knew they'd catch hell if they didn't, probably the reviewers too.
They used monkey and human cells.
Okay, yeah.
I would use a mouse cell or a rat cell of some kind,
Yeah.
right?
Yep.
But maybe they're assuming they were generalists, these
Yes.
rodent coronaviruses. So 97% of the positive rodents were from hotels and passenger stations. Only one was from a bamboo rat farm.
That's where the food is. I mean,
Yes.
So they had
where
35
the food-
samples from the farm and 35 samples from the populated places. So I would like you to speculate as to why the rats were more positive in the populated places. What's going on there, folks? I don't have any idea why that would be.
Dead silence.
I mean, they're not getting them from people, right? These are rodent viruses.
That's right.
So what?
Well, you could have a lot of exchange if you're at a rail station for rats that get on board and then get off at the local stops where their families live.
So maybe it's
so
like
you can have a lot of intercourse, a lot of social intercourse among the rat
it is a
population.
international destination. That's right. So
Yes.
rats are moving in and out. Yeah, I don't know
The rats.
why.
Well, no, the rats. The rats in a city may have larger colonies. They may, as Dixon said, have some degree of contact between the colonies that would spread viruses more easily.
Yeah.
Whereas in rural areas, you might have a smaller colony. And bamboo rats, being a totally different species, and these are the food rats. So I don't think that Norvegicus is usually used as a food by anybody. I could be wrong. I mean, starving people maybe, but it's not the same thing that people would be raising for food.
You would have to see the movie King Rat to get a good sense of that one.
Well, there's also a classical, it's kind of a quasi-mythological thing called a rat king, which was associated with terrible luck. It's supposedly if a group of rats get their tails tangled together, you end up with this multi-rat thing that it doesn't actually happen. But the theory is that this then brings down a curse on a town, and this is cropped up in myths and legends.
Like a syncytia of rats.
Yeah, it's kind of like you can look up a rat king, and it figures prominently in the plots of some fiction as well.
And there's the movie Ratatouille, of course, which is
Oh, right. Yes, that's a bit different.
one of my all-time favorites.
So they got full near or full genome sequence for 3 of these, and they give them very long names, which I will. They're rodent coronavirus, RCOV 3, 4, and 5. 3 and 5 were similar to known coronaviruses. 4 very low similarities, so they think it's a novel COV. But the number 3 was 96% similar to the China Rattus coronavirus HKU24. And number 5 was 97% identical to the Lucheng RN rat coronavirus, LRNV, which is a recombinant identified in China. I'm sure.
And recombinant, we don't mean it came from a lab.
Did not come from a lab.
This is a very common thing in coronaviruses. They recombine.
Number 3 and number 4 belong to the beta coronavirus genus, lineage A, and number 5 belongs to the alpha coronavirus genus. And if you are wondering what's this alpha and beta coronavirus business, so beta coronavirus genus contains lineage A also contains OC43 and HKU1. So
there's also Gamma and Delta.
Yeah.
And where were the taxonomists when they were naming the variants of concern
Yes.
Delta relative to this?
Apparently not
So
at the
now you
meeting.
have a beta coronavirus called Delta. Yes.
It's
Yeah.
lovely. It's lovely. Lovely. And then they, as Kathy said, in which we don't need to really discuss in any detail, they do some analysis to look for recombination, evidence of recombination. In other words, you see different origins of sequences in the genomes, and they find that this number 4 looks to be recombinant of MHV mouse hepatitis virus and this LRLV virus, which is interesting, which is consistent with this idea that these viruses recombine quite prolifically. So that's pretty much it. I think you can find coronaviruses in rodents. Urban sites in this particular study have a lot of them. One thing we don't know is the pandemic risk or the zoonotic risk of these viruses, right? And I think an interesting experiment would be to take the spike sequence of these viruses and put them into, say, WIV1 and see if it can, or a pseudotype, and see if it can infect human cells. And yes, that would be a gain of function experiment, but
Sure.
it's fine. We need to know, because otherwise, I mean, these viruses may not be able to infect human cells, in which case we should worry less about them. But if they did, then they
You'd
could
want to
be.
do that experiment under BSL-3 as a precaution.
I think so,
but
yes. I think so.
I think the risk would be pretty low.
But they do say that this shows that rodents which live near humans or can live near humans have the potential, have coronaviruses that have the potential to infect humans. So we should keep looking. We should do more surveillance. And as Kathy said, this is a One Health approach. Focus on the human, livestock, wildlife interface. Look at what viruses are there. And
International collaboration, exactly the kind of thing that needs to be done if we really want to find out the origins, and the kind of thing that is being roundly bashed by some in our political realm. Okay. Do you want the real answer or not, folks?
I think the answer is or not for a lot of them. Yeah. But in addition to finding the origins of SARS-CoV-2, which would be very interesting in a lot of ways, it's finding the origins of the next potential pandemic virus, which is really where we want to be so that we don't end up in the same situation in a few more years.
Exactly right.
You could almost hear Proxmire of however many 30 years ago or so.
Right, the golden fleece.
Saying, why are we studying viruses in rats?
Right.
You know, who cares?
Right.
Well,
yeah, that was the Golden Fleece Award. Is that right, Kathy?
I think that's true.
It was. That's
Yeah,
exactly
they used to bash stuff like Drosophila experiments.
Oh yeah, that was one of his favorites.
well, didn't the recent vice presidential candidate bash Drosophila experiments? Do you remember that, folks?
I don't remember that one specifically, but
A vice presidential candidate that was a former governor of Alaska. She said, I don't know why we're doing
Yes.
these experiments on flies and worms.
Yes, that's right.
Flies and worms, because eventually you'll end up with them.
Yes.
on that note,
Kathy, you said that one of these had a furin cleavage site, right?
I did not talk about the furin cleavage site, also known as the polybasic
cleavage site. Yeah, so our number 4, the number 4 isolate has RARR within the S protein. So another example of a polybasic cleavage site among coronaviruses, which adds to this, takes away from the smoking gun idea, right, folks?
Well, and that's another bad cleavage site. RAAR is not the canonical.
Yeah.
Right. So that's kind of a coronavirus take on a furin cleavage site.
All right. Let's do some picks of the week. Dixon Despommier, what do you have for us?
Vincent has muted for
You
us.
have your mute button.
I was being polite. You didn't want to hear me cough. So I've always had an attraction, let's say, that started when I was in high school for literature dealing with the unknown depths of the oceans.
I thought you were going to say a crush, and that would have been so perfect.
Well, there is a kind of crush if you go too deep. The first book I think I ever read from cover to cover as a freshman in high school, besides the literature that we were obliged to read, was a book by William Beebe called Half Mile Deep. And he was part of the Explorers Club in Bimini, and he was looking at the Bimini Trench through a bathysphere, which had about the size of a baseball opening with pure quartz as a window. That's all he could see throughout this bathysphere, right? And he would name these odd fish that he would see at the depths of 1,000 feet or 2,000 feet. And we don't know anything about this. I mean, we know more about the surface of Venus than we do about the depths of our own ocean. So there's a couple of books that just came out that were reviewed in the New York Times Sunday magazine section, the book section of that publication, that reviewed 2 books on the abyss. One of them was written by an explorer, and the other was written by this woman who's got a very appropriate name, Helen Scales.
Yes.
Because she's been attracted to fish and that sort of thing in the abyss as well. But what she's really concerned about is the fact that it's now open for economic exploitation.
Yeah.
And there are 2 big issues that she points out. One is the exotic fish industry. And the poster child for that is the orange roughy. And the orange roughy is found over a big, gigantic seamount off the coast of New Zealand. One place in the entire earth. And the orange roughy are eaten by giant squid. That's their food, at least in that area. And we knew that because if you find the dead sperm whale washed up on the beach somewhere in the South Island of New Zealand, its stomach is filled with parts of squid, and every now and then an orange roughy. So they knew there was a connection here somewhere. So the next thing you do, they used to call them slime fish because the deep trawlers used to pull them up and throw them back because they didn't think anybody wanted them, until finally somebody ate one. It's the most delicious saltwater fish you ever want to eat. And so we've extincted them, basically. They're almost gone. I don't know what that's doing for the giant squid industry or the sperm whales, but certainly there's a missing link here someplace. So that's one story that she talks about in the book. I'm talking about this book as though I've read it. I have not. I'm making this up from the review that I read of the book. I bought this book sight unread, and I'm going to read it from cover to cover because it covers an area that I'm very interested in.
Cool.
You know, she also talks about deep sea mining. You know, these chondrules that they find on the bottom of the ocean for magnesium and stuff like this, and manganese. It's easy mining, but it destroys entire ecosystems in the process. So we're good at that. We're good at destroying ecosystems. So I just thought there is no place left untouched by the heavy hand of human exploitation. And that's the basic bottom line on this one. Bottom, very, very bottom line.
Vincent, is this book illustrated?
I don't know that. I looked for in the review. I couldn't find it. I looked at it online on Amazon, and I couldn't find an illustration reference. So I don't know.
Dixon, according to the Australian Fisheries Management Authority, they can still capture roughy there.
Of course they can. Of course they can.
You said
It's
it was
not quite extinct.
It's not quite extinct.
Well, soon. Watch this space.
Yeah.
So it's.
Soon
I mean,
to be.
there are different areas of Australia. You know, the eastern, the western, the southern.
Yeah. But
And they have
they're over
different. It says not subject to overfishing in some areas, but in other areas uncertain. So,
all right.
I should not eat it anymore, right? That's the bottom line.
Or do you eat orange roughy? Do you like it?
Well, I've had it in the past. It's delicious. Yeah, at
It is,
restaurants.
isn't it?
Yeah, at
It's
restaurants.
very, very, very good. Very good.
It's really good. Excellent. All right, Kathy, what do you have for us?
Well, it's not all that creative for me. It's an astronomy picture of the day from Wednesday, but this one has a pretty cool animation. It's a solar system ball drop.
I love this.
They
This is
answer
great.
the question of whether a ball drops faster on Earth, Jupiter, or
good.
Uranus,
Oh, good.
and they show it for you. First of all, they show the sun and how fast.
Forget about that one.
That it's related to gravity, but also the distance from the center of gravity. And so you get some surprising results because, for instance, Uranus is cloudier or fluffier is another way of thinking.
Right. Less dense.
So it's pretty cool.
And the way they illustrate it is brilliant, where it's all the balls drop. They've got all the planets and the moon lined up, and all the balls drop simultaneously, and you see them fall to the sun and then just like down to the others of their own.
Very good. I like that.
I love it. I love it. That's great.
That's
Galileo would have loved it too.
Yes.
Rich, what do you have for us?
I have a phenomenon called the Dr. Fox effect that I thought was relevant to today's show, because we talked earlier about how people who don't know anything talk gobbledygook, and it results in misinformation that people assimilate. I had learned about this. I was thinking about it the other day because I'd heard reference to this years ago about some experiment involving somebody lecturing gobbledygook and seeing whether people believed it. And so I looked it up in my favorite resource, of course, Wikipedia, and found, sure enough, the Dr. Fox effect is a legitimate experiment conducted at the University of Southern California School of Medicine in the 1970s, where they had two speakers give lectures that are complete nonsense on the topic, quote, mathematical game theory as applied to physician education, okay? And it's total nonsense. And they had a scientist just deliver this, as just a regular lecture. And then they had an actor, and they did this to a couple of different groups, an actor do it kind of deadpan, and then do it, all, hyped up and with all sorts of, mannerisms and illustrations and stuff. And then they tested the students afterwards to see how much they got out of this and how they liked it. And of course, the animated, hyped up lecture got great ratings, and they seem to retain more of this nonsense than otherwise. And to me, the most amusing thing that I discovered in looking at this is that the actor involved was none other than Michael Fox. Okay, which is why it's called the Dr. Fox effect. Must have been before he hit the big time. Otherwise, everybody would have recognized
This is not Michael J. Fox.
Yeah.
It's not?
No.
Oh, I thought it was Michael J. Different.
This is an older Fox.
Yeah.
Myron Melvin Fox. Okay, great. Well, that's good.
Yeah.
At any rate, that's the story.
Nice.
Very cool.
I'd never heard of that. That's interesting.
And so your student evaluations are based more on flash than on content.
Yeah, I used to see this all the time in teaching, and it would drive me crazy. There were, a couple of teachers who, always got absolutely the highest ratings. But if you looked at the content of their lectures, it was not necessarily, thin.
So what accounts for the bad reviews? No flash? Less impressive presentation, but they found that even when presented in monotone, the students get learning from the real professor gain more knowledge subjectively tested. Alan, what do you have for us?
Well, Vincent, I know you like cockpit videos.
I do.
So this one is a little different from the airline cockpit videos that you're accustomed to. It was done last year. Patty Wagstaff is an air show pilot. So now air show aerobatic performers are, they perform a series of maneuvers that are very fast, often very violent in terms of shifts in G-force, much more aggressive in a lot of respects than anything any fighter pilot does. These are, in my opinion, some of the best pilots in the world. In order to be able to pull this off, they're often doing it at very low altitude to please the crowd. And there's And Patty Wagstaff is one of the best. She's a legendary airshow performer, spectacular pilot. And so in 2020, her shows got canceled because of the pandemic. But she needed to stay and practice. And she explains in this video, this is her workout. She's just going up at her local airport because she needs to maintain her G tolerance. Because the maneuvers that she does in her airshow routine are so extreme that if she doesn't maintain her tolerance to positive and negative Gs, she'll black out, which is obviously a big problem when you're flying, like this. So this is just her, her workout to maintain her G tolerance. And there's no other real way to do this besides to fly an airshow type routine, which she does in the aerobatic box that they have next to the airport she flies out of. And you get the cockpit view and it's looking at her, but it's the extra 300 has this bubble canopy for visibility. So you see this nice view of everything behind her and you see the kinds of maneuvers she's doing while she's casually monologuing about this. Okay, now we're going to do a couple of loops. Yeah. Now we're going to do a snap roll. I would have blacked out probably about 30 seconds after her takeoff. So this is just...
I mean, immediately after takeoff, she starts just spinning around.
Snap rolls off to the right to get to the box. And
oh my gosh.
Do you know where she's flying out of, Alan?
This is in Florida. I think she's in St. Augustine.
Yeah, I have a feeling I'm aware of this person because I think she flies out of near Amelia Island.
Okay.
I need to look at.
She's basically she's a in addition to flying air shows, she teaches aerobatics. And there's I am aware of this because it's something I've looked into occasionally. There's somebody much more convenient for me, a guy named Michael Goulian who flies in Massachusetts, who's of similar caliber, who I might.
What's this, a single engine plane now?
It is a small single engine plane that is built for exactly this.
It's got a big glass canopy, right?
It's basically an engine with some wings attached and a little tiny cockpit that she sits in with like the world's most uncomfortable seats, except that you're wearing a parachute when you strap into it because that's required for this type of thing. And it's like 300 horsepower pulling this.
Looks like she's got a GoPro on the front.
She's got a GoPro on the front that's shooting it. And this is her 2-seat plane that she uses for instruction. This is not her show airplane. So this is...
So Alan, before I would black out, I would die of a heart attack.
She has nice Bose headset there too. Yes, nice headphones and mic. Very nice.
That's a good quality.
I've had some correspondence with one of our listeners who's a pilot who at one point educated me about the relationship between height and being able to do this.
Yes.
And he says a lot of pilots are short because if you're too tall, the distance between your heart and your brain is big enough so that you black out with G forces.
Cool.
And G tolerance is another factor that she's training for in this case.
Can you make yourself G tolerant if you're not to begin with?
Being generally physically fit helps a lot.
Yeah.
But as she points out in the video, you can't if you're just lifting weights and doing the machines at the gym, you're not going to get the G tolerance because it's a specific thing that needs to be trained.
Do they do this with astronauts too?
G tolerance? I don't know. Now NASA has the equipment to just like put people in a centrifuge and give it to them that way. They wouldn't have to. But astronauts also do a lot of flying.
Yes.
Very cool. All right.
Very cool.
My pick is a YouTube channel called Office Hours. Well, actually, it's Alex Lindsay's channel, but the show is called Office Hours, where every day they live stream. They have like 20, 30 people on the Zoom call, and they join from all over the world. They talk about video and audio production, so I'm interested in it for that reason. But they also use Zoom to do this, and they get really good quality Zoom. And I've been trying to learn how they do it. There's no circuit diagram anywhere. And I once went to him, Alex Lindsay, on Twitter, and I said, "Can you give me a circuit diagram?" And his assistant answered and said, "Oh, just join the office hour." So basically they start at 6 a.m. and they go they record, I think, for an hour or two with all these people answering questions. And then they go offline for the rest of the day. It's basically 24/7 of people going there and answering questions. And it's 24 hours, 7-day-a-week thing on this particular, but they do different topics they often do focus on education, how to use video and audio in education and so forth. So they have some education specialists who will come on and answer questions. So you can actually, there's a link on the page, you can actually get a Zoom link and join them if you want to actually be there and ask questions. And then they only record like an hour or two of each day, and then they keep going all day. So I think it's an interesting concept that you could do this in high quality and teach people as well. So it's pretty cool. They also do cooking on the weekends. They get people who will put cameras in their kitchens and they teach you how to cook. But it's all with this technology where sometimes they have 20 people on the Zoom, right? And they're all really good quality because they're streaming it that way and they've got it set up. So it's 2 hours each, and then the rest of the day, if you could just pop in and out anytime. Office hours. Cool. Which is not really what office hours is. No. It's not all day, folks. So. We have 2 listener picks. Karen, dear esteemed TWiV team, I think you will enjoy this episode of Malcolm Gladwell's Revisionist History Podcast if you have not listened already. Dogs are amazing creatures, as I'm sure the dog lovers on the team know. At $2.25 per COVID-19 test for 2 dogs, handlers, and training, it's a bargain. Wow. That's cheap. It's almost a lick-a-stick. Thank you for all you do. I listen to every episode, even though much is way over my pay grade. Karen is in sunny Calgary. So this is an episode called The Dog Will See You Now. Malcolm Gladwell, apparently who likes TWiV. He has mentioned multiple times on various pods. People have sent in that he really likes TWiV. Okay, thank you, Malcolm. That's cool. I'm going to get a COVID test and do it on myself. The hell with the dog. For $2.25. I wonder what it is.
That's for the dog test.
Yeah. I mean, you could use, it's probably just a swab. What the heck, right? What is it at COVID?
Wait, no, I think are these COVID sniffing dogs?
I think so.
No,
I think that's what's being done here. It costs you $2.25 to hire these people per test to hire them to do this, right?
Oh, really? It's for dogs sniffing it out? Oh, okay.
Yeah. It refers to 2 dogs, handlers, and training.
I thought it was to test your dog. Too bad.
I haven't listened to the podcast yet, but I'm just basing this on the email.
Well, that's interesting also. You can, I believe that dogs can sniff out the volatile organics that are produced when you get infected.
Yeah.
And Alicia writes, hello, TWiV team. I'm writing to you from Ontario, Canada. Follow up to Brienne's pick from episode 796 of the PCR song. If you think that one got stuck in your head, have a listen to the one my grad school advisor showed our lab while discussing PCR techniques during lab meeting, also created by BioRad. YouTube link. Thank you.
This I actually watched. This is good.
This is funny.
Thank you for always being a reliable source of information throughout this pandemic. All right. Thank you for your.
By the way, Patty Wagner flies out of St. Augustine.
Yeah, Patty Wagner. Right. And I just wanted to mention one more thing about the video. This virtual air show thing, part of the gimmick of it is that it's in the upper left corner, there's a little D-pad for directional control that you can click on the YouTube video and pan the view around. So if you would prefer the front view out of the airplane.
Oh, I was going to ask about that.
Yeah.
Cool.
Which will not reduce the effect if you're subject to motion sickness.
Vertigo. That's right.
Yeah. But I did feel like I wanted to have the front view.
You wanted to see, yeah.
Yeah.
Yeah, so you can see, you can look back at her as she's monologuing, or you can turn around and see the forward view as she's doing the maneuvers.
Right.
Nice.
So I guess another thing I wanted to say was that I was alerted to the fact that Brianne Barker was on the Brian Lear show, which is an NPR radio show, a couple of mornings ago, and I listened to it. She was wonderful.
Yeah,
she was absolutely spectacular. When she didn't know something, she said, "I don't know that." And when she knew it, she gave a great, really, really good, solid, easy to understand explanation. So I emailed her, of course, after that, and she thanked me for it. But she was very good. Very good.
Cool.
Brian Lear, unfortunately, wasn't hosting the show that day. There was a stand-in for him. I think he was out reacting to the Afghanistan tragedy and getting his guest lines up. But she was great. So that was one of us. And TWiV was mentioned right up front.
Good.
So we got a good shout-out.
All right. TWiV 798 up. Looks like Daniel Griffin's going to get number 800. I thought of canceling Tuesday, but I said, no, Daniel could use number 800. That would be fun for him.
He would like that.
We'll take 1,000, okay?
At which point you said we would have another celebration. At 500, you said we wouldn't
we
do
were
anything
promised a
again until
celebration at
1,000.
1,000.
Yeah.
God, it came quickly, didn't it?
Yeah.
It did.
Careful what you promise.
Yeah.
That's
I don't
right.
mind. No, we'll do something. So let's see.
TWiV 1000 live from Wuhan.
It's going to be in, what are we doing?
That might be pushing it.
We're doing 100 a year now, right?
Yeah.
Yeah.
So 2 years. Yeah.
If we keep up at this pace post-pandemic.
I can't imagine we're going to be doing 2 a week for, God, I hope not. I mean, the only reason I say, God, I hope not, is that if we're doing 2
means the
a
pandemic
week,
is still going.
Yeah.
Yeah, it means things are still bad.
Well, one thing we could do is all meet at the incubator and do it there and have a small audience, of special.
That's a good idea.
That'd be good.
And I'd fly everybody in, right? Then
Cool.
that could be fun. Alan, you're not too far away. Dixon, you're
No, I'm
not far away.
at all.
Now I can almost walk.
All right, 798 microbe.tv/TWiV for the show notes. Send us your questions and comments, TWiV@microbe.tv. If you like what we do, consider supporting us, microbe.tv/contribute. Dixon DePalmier is at trichinella.org, thelivingriver.com. Thanks, Dixon.
You're welcome, Vincent, and everybody else. Glad to have the regulars back in their seats. Good to see Kathy again.
You haven't seen Kathy in a while, have you, Dixon?
No, I have not. I have not, except by looking at special TWiVs. I have not seen her in person.
Rich Condit, Emeritus Professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Alan Dove, alandove.com, Alan Dove on Twitter. Thanks, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV and Ronald Jenkins for their music. I also want to thank all of you contributors who have been giving for the past years over at PayPal and Patreon, and then a few of those that send me paper checks that I figure out what to do with every now and then. Elio Schechter sent one. He wrote it out to This Week in Microbiology. I said, no, I can't cash that. You got to make it out to micro. So I sent it back to him. But he had originally sent it to This Week in Microbiology, Columbia University, New York. And it ended up way downtown.
Dear.
And they called me and said, they opened it, actually. We have a check for you. So it took many months to get there. And then they put it in Campus Meal. It took another month to get up
Of course
here.
it did.
And then I said, well, I have to send it back to Elio because I can't cash it. So he sent me a new one. Thank you all for your support. It's really, really important. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.